Greetings, my name is Declan, and I'm here to discuss a recent breakthrough in the field of gene therapy. But firstly, some background. First off, what is gene therapy? Gene therapy is the use of a gene or genes to treat or prevent a disease or medical disorder. The gene therapy, the process is achieved by adding new copies of a gene to a broken gene or by replacing an entirely defective or missing gene in a patient's cell with a healthy version of that cell, usually delivered in virus form. Gene therapy was first proposed as a treatment for genetic diseases and disorders in 1972 on the 3rd of March at the Salk Institute in San Diego, USA. The first successful use of gene therapy was performed by an American physician by the name of William French Anderson at the National Institute of Health on a 4-year-old girl who suffered from combined immunodeficiency, better known as SCID. In the modern day, gene therapy has been used to treat conditions such as certain inherited disorders and diseases like cystic fibrosis, and sickle cell disease, and even some cancers and blindness caused by retinitis pigmentosa. But back on topic. So what condition is the new breakthrough affecting, and how useful can it be? First, we need to understand some basic medical terminology. The condition causes an irregular heartbeat, better known as an arrhythmia. An arrhythmia can be caused by a range of medical conditions, and from more on point to our discussion today, it can be caused by genetics. The condition is CPVT. I will not try to say its extended name because we'll be here all day. And it is caused by a mutation in the cardiac rhyodine receptors, RYR2, and the cardiac calsequestrin, or CASQ2, that results in an irregular heartbeat from the loss of two proteins associated with triadenine, TRD, and junctin, JNC, which is responsible for the regulation of the macromolecular complex devoted to controlling of the calcium release in the heart area, and that is responsible for the muscle contraction and relaxation, causing it to cause an irregular heartbeat. The innovation itself was conducted by researchers at the Boston Children's Hospital and Harvard Stem Cell Institute, and they have created the first human tissue models of the condition in CPVT. This model provided a proof of concept that CPVT could be effectively treated by gene therapy using the AVV9-GFP gene therapy method. So how did this come to be? Well, CPVT is found in 1 in 10,000 people, and is roughly have a mortality rate of roughly a third by the age of 30 years. This means that a large portion of the population currently lives with CPVT, and many of these people do not have a cure. And the most effective treatment is a radical change and not experience many of the thrills of life, as rushes of adrenaline can cause the arrhythmia to be increased on an order of magnitude. A cure to such a condition would be greatly appreciated by many of these people, obviously those that would be cured of the disease, and for the greater world of medicine, as it would be a massive step for gene therapy. Other effective treatments are beta blocker medication to block adrenaline on the heart, or one of two forms of defibrillators, an implanted cardioverter in the body itself, or an automatic external defibrillator to deliver an electric shock to restore normal heart rhythm. Currently, gene therapy for CPVT has only been tested on small animals, such as mice, and in human models of the condition, basically simulations. So is this innovation reliable and/or valid? That can be debated. The project used many of the proven techniques used throughout many other innovations and projects in the biotech field, such as the human cell models, which themselves are innovation. As the innovation is a stepping stone towards a practical solution from a simulation in theory, it itself does not have to promise a breakthrough, but lay a path towards one. But what is their strength is also their weakness. The human cell models aren't a human cell. They are far less complicated / specialized and do not exactly replicate a human cell. And as the current project only used 2 models of 2 different human cells, it doesn't really allow for much change. So moving forward, what are some possible recommendations? Obviously, putting more cell models into the report and increasing their diversity. But this does prove a pathway into human trials and into practice.